So adula vandu DNA la pathanga some bacteria is very good for us, some adala which we can find. So this particular book tries to explain. Ipo pathanga unnoru book vandu Life a Science of Biology by Sadava. Idu vandu life can we now explain it as a study of molecules themselves which act together. So, for example, if we spoil the environment, it is not good for the next generation. We know that. So, how to do this whole about the whole of science? We have to biology. We have to think in a very holistic manner. There is another for one advanced level in the website www.ioibioscience.org. For example, what is great about biology? We thought that biology was not a quantitative subject, right? Biology. So biology can also be put under firm foundations like chemistry, physics, and mathematics. So a lot of physicists, chemists, and mathematicians are getting into biology. So their views on biology is kept here. So in the why was the Nobel Prize given?
Placels.
How our brain cells are there at different locations. But chemistry what was the prize given for? Microscopy. Okay. இப்போ வந்துட்டு நீங்க மைக்ரோஸ்கோபி பார்த்தீங்கன்னா ஒரு மைக்ரோஸ்கோப்பை பார்த்தீங்கன்னா அதோட லிமிடேஷன் என்ன? பிசிசிஸ்ட் ஓட லிமிடேஷன் என்னது? இப்போ லைட் இஸ் மேட் அப் ஆஃப் photons or also they are waves, right? So இப்போ நான் இதெல்லாம் பார்க்கணும்னா அந்த wavelength ஓட சைஸ்ல தான் பார்க்க முடியும். அதுக்கு கீழே பார்க்க முடியாது இல்லையா? லைட் மைக்ரோஸ்கோப்பி யூஸ் பண்ணிட்டு I cannot use anything less than the wavelength of. So this is called the Abbe's limit. That is lambda by 2. Lambda by 2 கீழே நான் என்னால என்னத்தையும் பார்க்க முடியாது. If I use but நம்ம electromagnetic radiation நம்மளுக்கு எல்லாருக்கும் தெரியும் இல்லையா? அந்த மாதிரி for example X-ray has shorter wavelength. So adu vechi paakalam. But then what will happen? X-ray will destroy the micro because of its high energy. So for the latest Nobel Prize was to go over the Abbe's limit. So adu they used a special technique called so it was actually done by a physicist and he won the Nobel Prize with 2 more. So the last book is an excellent book. It's the Molecules of Life, Physical and Chemical Principles by Puthanampatti, Wemmer and Kurian. This is actually, they are all physicists who wrote this book. And most of my slides are taken from this particular book. So, the title of the talk that I'm going to give today is a chemist's rather limited view of biology. If you think the most important molecule in biology is what do you think is the most important molecule in biology? DNA, right? So the most important, because without DNA, we don't have the blueprint of life. So we, chemist view of it, nature chose DNA. We try to give an explanation. So we will start off with this. So it's a bacteria, right? So bacteria, there are some tails hanging around. Flagella. So why is there a flagella in the bacteria? So how does flagella help in locomotion? How does flagella flagella every locomotion of a bacteria.
It pushes it forward.
But the flagella is so wavy, so you can't use it as an oar. Oar has to be stiff. Correct. So yeah, நம்ம boatல oar மாதிரி அதை யூஸ் பண்ண முடியாது. Because அது வந்து stiff-ஆ இருக்கும். Oar is not stiff. It's very wavy. So we will try to explain this. So luckily by microscopy we can see part of this cell. So இப்போ பார்த்தீங்கன்னா இதுதான் இந்த part வந்து நம்ம expand பண்ணிருக்கோம். We have expanded that part here. Correct? So this is where the flagella goes inside the cell. So the flagella, this goes inside the cell like that. This goes like that. The flagella. Right. So let us now try to understand this by some kind of whether mechanical device. Does this remind you of any mechanical device? Does this remind you of screw? Anything else?
Lever.
Yeah, lever mari that's not a mechanical device. It's fixed. Mechanical device is something that is, we will define crudely a mechanical device as something that is capable of motion. Okay, right? Very good. Gear. Correct. So this resembles a gear. So the teeth are right? This is the teeth mari irukku. So gear la the teeth irukum. So there's another gear like this. So this is on that, right? So what kind of motion is regulated by the gear? Rotate. Very good. So rotational motion. So now how does the bacteria move? Rotates the flagella. Rotates it very fast. Why do you need the gear? For making it stop or making it go in some speed, at some speed. So now we will know that the flagella is there for locomotion. Neenga da correct ah sonnunga. And that locomotion is by rotating the flagella at speeds. So for the rotate another, what does it need for rotation? It needs energy. Correct. So what molecule gives energy? ATP. So so you have ATP synthase. It synthesizes ATP and gives it. Right. So you see this. Now, how to make the flagella, how to make the whole cell, right? We have to have a book, right, to tell us or tell nature how to make the bacteria. So on the book is this. This is the DNA. The DNA contains the whole book. Well, DNA is made up of what molecules? Nucleic acids. Correct. So for nucleic acids is this, but on the nucleic acids are very stiff, right? So we need something else. If you ஒரு கார் பண்ணனும்னு வெச்சுக்கங்க, right? அதுக்கு ஒரு book இருக்கு. That is the manual of the how to make a car. But you cannot make the car out of paper. You have to make it out of iron, aluminum, plastic, etc., etc. So the building blocks of the car are different from the building blocks of the manual. So the manual is written in nucleic acid. The bacteria is made up of protein. Okay. So அது நீங்க பார்த்தீங்கன்னா இப்போ நான் flagella தான் இப்போ ஒரு கார்ல steering wheel மாத்த தான் பண்ணணும். So something happens to the steering wheel. I have to only. So what should I do? I should not be in a position that I have to read the whole book to find out how to make the steering. So what should I do? Chapters, right? I should arrange it in chapters. So Indian DNA it is arranged in chapters. So every cell has a DNA. Only some parts of the DNA will be read at a time. So that is done by what is known as a RNA. Right? The RNA only reads specific parts, translates it into protein and makes the flagella. So This is basically the mechanical aspect of locomotion, of making how to explain cellular function using mechanical thought. So this is something that all of you know, yeah. So this is the first is replication. So now first, when we have conceived, right? How many DNA molecules do we have? When we were conceived, how many DNA molecules do we have? First, right? A sperm and an egg meet each other, right? And make a cell, one cell. So how many DNA molecules will we have? Two, right? So one strand from the mother, one strand from the father, right? So we make, so if when we are first conceived, we have one DNA molecule. How many DNA molecules do we have now? So the number of DNA molecules is going to be equal to the number of cells we have, right? So how many cells do we have now? Countless. Countless, then we should be infinity, right? We can't count it. It is actually 1 trillion. Okay, we have 1 trillion cells. So we have 1 trillion DNA molecules inside us. So within about 9 months of gestation, from one cell we become 1 trillion cells. So that process is called replication. So all of us will have the complete cell in our body will have the complete DNA of us. Okay. But immediately you would have seen the skin cell is different from the brain cell. Right? Pictures. Brain cell is like a star shape. It's like a sort of a rhombus, which is skewed rhombus. Skin cell, it will be spherical. Right? So what does that tell you? Okay, but what does it tell you from a DNA perspective? Not all the DNA is used to make a cell. Only part of the DNA is used to make the cell, right? Because otherwise, if we use the whole DNA, what will happen? All cells should be the same, and we should have the function should be the same. But the very fact that we have different cells tells us that in the money only some parts of the DNA are taken and then read to make that particular cell. So this is the reading is done by some enzymes, and then on the read is transcribed into what is known as an RNA molecule. Right? So அந்த சாப்டர் தான் நம்ம படிச்சு அந்த சாப்டரோட அப்படியே அந்த சாப்டர் மாத்திரம் எடுக்கிற மாதிரி, right? அந்த சாப்டர் மாத்திரம் எடுத்தா இந்த சாப்டர் will make a brain cell அப்படின்ட்டு நமக்கு தெரியும். ஸோ அந்த சாப்டர் மாத்திரம் எடுத்துட்டு then what do? We put it into a translation, right? நம்ம சொன்னோம் இல்லையா இந்த மாதிரி that the nucleic acids which are present in RNA are very rigid. So we want something. So அதை நம்ம protein translate பண்ணணும். So ஏன் translation அது? இதோட language nucleic acid. Protein ஓட RNA ஓட alphabet nucleic acid. Protein ஓட alphabet amino acid. So alphabets of proteins are amino acids. Alphabets of RNA are nucleic acids. So அதை நம்ம translate பண்ணணும். That is, from a nucleic acid alphabet, we should get it into a amino acid alphabet, right? So that is done by a machine. That's called what is it called? The translation of RNA to protein is done by a small machine, right? And the machine is ribosome, right? ribose. So you will see that that is the ribose. And why is the you would have seen that 4 years ago who won the Nobel Prize for Venki Ramakrishnan. Right? Very good. Excellent. So, first is quite long. So, so let us now concentrate on why nature chose DNA. For DNA, and the DNA is made up of 3 parts. One is the base, second is the sugar, and third is the phosphoric acid. So, we have the base, sugar, and phosphoric acid. So, first we must know what are these bases. How many bases are there? 4 bases. What are the bases called? Adenine, guanine, thymine, and cytosine for DNA. So this is the structure of the bases. So there are broadly 2 kinds of bases, right? One is a 6-membered ring fused to a 5-membered ring. One is a 6-membered ring. Yeah. One is a 6-membered ring fused to a 5-membered ring. Other is just a 6-membered ring. So in the pair of the purine. The purine level you have adenine and guanine. In pyrimidine you have cytosine and thymine. Cytosine and uracil in the term of in RNA. Okay. So you pair first to base. Why do we call this a base? First we must understand why we call this a base. Phosphoric. Base, deoxyribose, and phosphoric. So why is this called a base? So what is our definition of a base? Very good. It accepts electrons, right? Does it accept electrons or does it give electrons? Base. Gives electrons, right? So, for example, why is ammonia called a base? Ammonia, there is no hydroxyl ion, right? But we still call it a base. Why do we call it a base? Because when you put it in water, right, there are lone pairs of electrons, and that takes the H plus from water and releases a OH minus. So you will see that anything that has lone pair of electrons that can accept, that can give electrons, we will call it a base. So then we can call this a base because there are lone electrons on this nitrogen, and this nitrogen, and therefore we call it a base. Now sugars yeah. So sugars, do you know about what kind of sugars do you know? Glucose and fructose. Right? So sucrose is made up of 1 molecule of glucose and 1 molecule of fructose. Correct. So the sugar would fructose is a 5-membered sugar. Glucose is a 6-membered sugar. So what shall we call them? For example, a 5-membered ring, what will you call it? Penta. So the pair of them we can call it pentose. So fructose is a pentose. Glucose is a hexose, right? So in the body, depending upon the arrangement of the OH groups, there can be different sugars. So one of the sugars that resembles fructose is called ribose, right? So this is a ribose. So ribose is a pentose. So the numbering pattern, you will see it as 1, 2, 3, 4 and 5. Right. So you can see that 1, 2, 3, 4 and 5. Correct. So you will see that ribose is a sugar, right? With a pentose. And if you would have heard of certain other names like dehydrate. What is dehydrate? Immersion of water. So deoxyna, removal of oxygen. So for ribose, ribose will have a OH here, right? So if I remove that oxygen, what should I call it? Deoxy. Right. That's why it is called deoxy. Right. That's where I remove that oxygen from the second position. So I will call it 2-deoxy. Right. Correct. So now this OH and this hydrogen can react. There can be a dehydration reaction, and therefore you can form a bond. So this is the first slide you told me about esterification, right? What is the esterification reaction? Alcohol and carboxylic acid. So that is basically an organic ester. Correct. So ademari, you can also see that you can form inorganic esters. So inorganic esters can we can take an alcohol and react it with mineral acid, right? So that's exactly what nature does. So this is an alcohol, yeah, CH2 reacts with phosphoric acid, which is a mineral